Biostatistics are the application of statistics to a wide range of topics in biology. It encompasses the design of biological experiments, especially in medicine, pharmacy, agriculture and fishery, the collection, summarization, and analysis of data from those experiments, and the interpretation of, and inference from, the results. A major branch is medical biostatistics, which is exclusively concerned with medicine and health. History. Biostatistics and genetics. Biostatistical modeling forms an important part of numerous modern biological theories. Genetics studies, since its beginning, used statistical concepts to understand observed experimental results. Some genetics scientists even contributed with statistical advances with the development of methods and tools. Gregor Mendel started the genetics studies investigating genetic segregation patterns in families of peas and used statistics to explain the collected data. In the early 1900s, after the rediscovery of Mendel's Mendelian inheritance work, there were gaps in understanding between genetics and evolutionary Darwinism. Francis Galton tried to expand Mendel's discoveries with human data and proposed a different model with fractions of the heredity coming from each ancestral composing an infinite series. He called this the theory of law of ancestral heredity. His ideas were strongly disagreed by William Bateson, who followed Mendel's conclusions that genetic inheritance were exclusively from the parents, half from each of them. This led to a vigorous debate between the biometricians, who supported Galton's ideas, as Walter Weldon, Arthur Dukenfield Derbyshire and Carl Pearson, and Mendelians, who supported Bateson's and Mendel's ideas, such as Charles Davenport and Wilhelm Johannsen. Later, biometricians could not reproduce Galton conclusions in different experiments, and Mendel's ideas prevailed. By the 1930s, models built on statistical reasoning had helped to resolve these differences and to produce the neo-Darwinian modern evolutionary synthesis. Solving these differences also allowed to define the concept of population genetics and brought together genetics and evolution. The three leading figures in the establishment of population genetics and this synthesis all relied on statistics and developed its use in biology. Ronald Fisher developed several basic statistical methods in support of his work studying the crop experiments at Rothamsted Research, including in his books Statistical Methods for Research Workers 1925 and The Genetical Theory of Natural Selection 1930. He gave many contributions to genetics and statistics. Some of them include the ANOVA, p-value concepts, Fisher's exact test and Fisher's equation for population dynamics. He is credited for the sentence natural selection as a mechanism for generating an exceedingly high degree of improbability. Sewell G. Wright developed F-statistics and methods of computing them and defined inbreeding coefficient. J. B. S. Haldane's book, The Causes of Evolution, re-established natural selection as the premier mechanism of evolution by explaining it in terms of the mathematical consequences of Mendelian genetics. Also developed the theory of primordial soup. These and other biostatisticians, mathematical biologists, and statistically inclined geneticists helped bring together evolutionary biology and genetics into a consistent, coherent whole that could begin to be quantitatively modeled. In parallel to this overall development, the pioneering work of Darcy Thompson in On Growth and Form also helped to add quantitative discipline to biological study. Despite the fundamental importance and frequent necessity of statistical reasoning, there may nonetheless have been a tendency among biologists to distrust or deprecate results which are not qualitatively apparent. One anecdote describes Thomas Hunt Morgan banning the Friedan calculator from his department at Caltech, saying, "Well, I am like a guy who is prospecting for gold along the banks of the Sacramento River in 1849. With a little intelligence, I can reach down and pick up big nuggets of gold. And as long as I can do that, I'm not going to let any people in my department waste scarce resources in placer mining." Topic: Biostatistics and Medicine. Statistical concepts also are present in clinical trials and epidemiological studies. These fields have their own history of biostatistics developments. In the 18th century, statistical methods were used to decide whether the application of certain treatments were effective, such as the insertion of smallpox pustules under an individual's skin in the hope of creating a mild case of the disease that would induce later immunity. Since this actually put patients at risk of contracting a potentially fatal form of the disease, this treatment became the subject of much controversy. John Arbuthnot in 1722 studied the chances of people dying by naturally occurring smallpox as compared to inoculation-induced smallpox. On the basis of the statistical studies, it was concluded that inoculation was preferred. Later, Daniel Bernoulli and Jean D'Alembert developed more robust statistical methods for the same problem, both based on the proportion of people who died. James Lind were the first to propose groups of test of hypotheses. He applied this method to solve an outbreak of scurvy. For this, he is considered the father of clinical trial. In 1835, Pierre-Charles Alexander Lewis proposed the numerical method to argue that the practice of bloodletting was actually doing more harm than good for the patients. In 1840, Louis Denis Jules Gavaret published the Principes généraux de statistique médicale, in which he pointed out that Lewis's averages could vary between what he called limits of oscillation or confidence interval if multiple samples were taken from the same population. Carl Pearson also expanded his methods to medicine, despite his main goal was to explicit the statistical implications of Darwin's theory of natural selection. Topic: Research planning. Any research in life sciences is proposed to answer a scientific question we might have. To answer this question with a high certainty, we need accurate results. The correct definition of the main hypothesis and the research plan will reduce errors while taking a decision in understanding a phenomenon. The research plan might include the research question, the hypothesis to be tested, the experimental design, data collection methods, data analysis perspectives and costs involved. It is essential to carry the study based on the three basic principles of experimental statistics: randomization, replication, and local control. Topic: Research question. The research question will define the objective of a study. The research will be headed by the question, so it needs to be concise. At the same time, it is focused on interesting and novel topics that may improve science and knowledge in that field. To define the way to ask the scientific question, an exhaustive literature review might be necessary. So, the research can be useful to add value to the scientific community. Topic: Hypothesis definition. Once the aim of the study is defined, the possible answers to the research question can be proposed, transforming this question into a hypothesis. The main propose is called null hypothesis H0 and is usually based on a permanent knowledge about the topic or an obvious occurrence of the phenomena, sustained by a deep literature review. We can say it is the standard expected answer for the data under the situation in test. In general, HO assumes no association between treatments. On the other hand, the alternative hypothesis is the denial of HO. It assumes some degree of association between the treatment and the outcome. Although, the hypothesis is sustained by question research and its expected and unexpected answers. As an example, consider groups of similar animals, mice, for example, under two different diet systems. The research question would be, what is the best diet? In this case, H0 would be that there is no difference between the two diets in mice metabolism H0, mu1 equals mu2, and the alternative hypothesis would be that the diets have different effects over animals' metabolism H1, mu1 does not equal mu2. The hypothesis is defined by the researcher, according to his/her interests in answering the main question. Besides that, the alternative hypothesis can be more than one hypothesis. It can assume not only differences across observed parameters, but their degree of differences, i.e. higher or shorter. Topic: Sampling. Usually, a study aims to understand an effect of a phenomenon over a population. In biology, a population is defined as all the individuals of a given species, in a specific area at a given time. In biostatistics, this concept is extended to a variety of collections possible of study. Although, in biostatistics, a population is not only the individuals, but the total of one specific component of their organisms, as the whole genome, or all the sperm cells, for animals, or the total leaf area, for a plant, for example. It is not possible to take the measures from all the elements of a population. Because of that, the sampling process is very important for statistical inference. Sampling is defined as to randomly get a representative part of the entire population, to make posterior inferences about the population. So, the sample might catch the most variability across a population. The sample size is determined by several things, since the scope of the research to the resources available. In clinical research, the trial type, as inferiority, equivalence, and superiority is a key in determining sample size. Topic: Experimental design. Experimental designs sustain those basic principles of experimental statistics. There are three basic experimental designs to randomly allocate treatments in all plots of the experiment. They are completely randomized design, randomized block design, and factorial designs. Treatments can be arranged in many ways inside the experiment. In agriculture, the correct experimental design is the root of a good study, and the arrangement of treatments within the study is essential because environment largely affects the plots, plants, livestock, microorganisms. These main arrangements can be found in the literature under the names of lattices, incomplete blocks, split plot, augmented blocks, and many others. All of the designs might include control plots, determined by the researcher, to provide an error estimation during inference. In clinical studies, the samples are usually smaller than in other biological studies, and in most cases, the environment effect can be controlled or measured. It is common to use randomized controlled clinical trials, where results are usually compared with observational study designs such as case control or cohort. Topic: Data collection. Data collection methods must be considered in research planning, because it highly influences the sample size and experimental design. Data collection varies according to type of data. For qualitative data, collection can be done with structured questionnaires or by observation, considering presence or intensity of disease, using score criterion to categorize levels of occurrence. For quantitative data, collection is done by measuring numerical information using instruments. In agriculture and biology studies, yield data and its components can be obtained by metric measures. However, pest and disease injuries in plants are obtained by observation, considering score scales for levels of damage. Especially, in genetic studies, modern methods for data collection in field and laboratory should be considered, as high-throughput platforms for phenotyping and genotyping. These tools allow bigger experiments, while turn possible evaluate many plots in lower time than a human-based only method for data collection. Finally, all data collected of interest must be stored in an organized data frame for further analysis. Topic: Analysis and data interpretation. Topic: Descriptive tools. Data can be represented through tables or graphical representation, such as line charts, bar charts, histograms, scatterplot. Also, measures of central tendency and variability can be very useful to describe an overview of the data. Follow some examples. Frequency table. Zone type of tables are the frequency table, which consists of data arranged in rows and columns, where the frequency is the number of occurrences or repetitions of data. Frequency can be absolute, represents the number of times that a determined value appear. N equals F1 plus F2 plus F3 plus F n display style n equals f underscore 1 plus f underscore 2 plus f underscore 3 plus f underscore n relative, obtained by the division of the absolute frequency by the total number n I equals f I n display style n underscore I equals frac f underscore I n. In the next example, we have the number of genes in 10 operons of the same organism. G E N E S equals 2 3 3 4 5 3 3 3 3 4 display style genes equals 2 3 3 4 5 3 3 3 3 4. Line graphs represent the variation of a value over another metric, such as time. In general, values are represented in the vertical axis, while the time variation is represented in the horizontal axis. Bar chart. A bar chart is a graph that shows categorical data as bars presenting heights vertical bar or widths horizontal bar proportional to represent values. Bar charts provide an image that could also be represented in a tabular format. In the bar chart example, we have the birth rate in Brazil for the December months from 2010 to 2016. The sharp fall in December 2016 reflects the outbreak of Zika virus in the birth rate in Brazil. Histograms. The histogram or frequency distribution is a graphical representation of a dataset tabulated and divided into uniform or non-uniform classes. It was first introduced by Carl Pearson. Scatter plot. A scatter plot is a mathematical diagram that uses Cartesian coordinates to display values of a dataset. A scatter plot shows the data as a set of points, each one presenting the value of one variable determining the position on the horizontal axis and another variable on the vertical axis. They are also called scatter graph, scatter chart, scattergram, or scatter diagram. Mean. The arithmetic mean is the sum of a collection of values x 1 plus x 2 plus x 3 plus x n. Display style x underscore 1 plus x underscore 2 plus x underscore 3 plus cdots plus x underscore n divided by the number of items of this collection n. Display style n x equals 1 n I equals 1 n x I equals x 1 plus x 2 plus x n n. Display style bar x equals frac 1 n left sum underscore I equals 1 caret n x underscore I right equals frac x underscore 1 plus x underscore 2 plus cdots plus x underscore n n. Median. The median is the value in the middle of a dataset. Mode. The mode is the value of a set of data that appears most often. Box plot. Box plot is a method for graphically depicting groups of numerical data. The maximum and minimum values are represented by the lines, and the interquartile range IQR represent 25 to 75% of the data. Outliers may be plotted as circles. Correlation coefficients. Although correlations between two different kinds of data could be inferred by graphs, such as scatter plot, is necessary validate this through numerical information. For this reason, correlation coefficients are required. They provide a numerical value that reflects the strength of an association. Pearson correlation coefficient. Pearson correlation coefficient is a measure of association between two variables, x and y. This coefficient, usually represented by ρ for the population and r for the sample, assumes values between -1 and 1, where ρ Topic 1 represents a perfect positive correlation, ρ -1 represents a perfect negative correlation, and ρ = 0 is no linear correlation. Topic: Inferential statistics. It is used to make inferences about an unknown population, by estimation and/or hypothesis testing. In other words, it is desirable to obtain parameters to describe the population of interest, but since the data is limited, it is necessary to make use of a representative sample in order to estimate them. With that, it is possible to test previously defined hypotheses and apply the conclusions to the entire population. The standard error of the mean is a measure of variability that is crucial to do inferences. Hypothesis testing is essential to make inferences about populations aiming to answer research questions, as settled in Research Planning section. Authors define 4 steps to be set. The hypothesis to be tested, as stated earlier, we have to work with the definition of a null hypothesis H0 that is going to be tested, and an alternative hypothesis. But they must be defined before the experiment implementation. Significance level and decision rule. A decision rule depends on the level of significance, or in other words, the acceptable error rate alpha. It is easier to think that we define a critical value that determines the statistical significance when a test statistic is compared with it. So, alpha also has to be predefined before the experiment. Experiment and statistical analysis. This is when the experiment is really implemented following the appropriate experimental design, data is collected and the more suitable statistical tests are evaluated. Inference is made when the null hypothesis is rejected or not rejected, based on the evidence that the comparison of p-values in alpha brings. It is pointed that the failure to reject H0 just means that there is not enough evidence to support its rejection, but not that this hypothesis is true. Confidence interval. A confidence interval is a range of values that can contain the true real parameter value in given a certain level of confidence. The first step is to estimate the best unbiased estimate of the population parameter. The upper value of the interval is obtained by the sum of this estimate with the multiplication between the standard error of the mean and the confidence level. The calculation of lower value is similar, but instead of a sum, a subtraction must be applied. Topic: Statistical considerations. Topic: Power and statistical error. When testing a hypothesis, there are 2 types of statistic errors possible: type 1 error and type 2 error. The type 1 error or false positive is the incorrect rejection of a true null hypothesis, and the type 2 error or false negative is the failure to reject a false null hypothesis. The significance level denoted by alpha is the type 1 error rate and should be chosen before performing the test. The type 2 error rate is denoted by beta, and statistical power of the test is 1 minus beta. Topic: P-value. The p-value is the probability of obtaining results as extreme as or more extreme than those observed, assuming the null hypothesis H0 is true. It is also called the calculated probability. It is common to confuse the p-value with the significance level alpha, but the alpha is a predefined threshold for calling significant results. If p is less than alpha, the null hypothesis H0 is rejected. Topic: Multiple testing. In multiple tests of the same hypothesis, the probability of the occurrence of false positives, family-wise error rate increase, and some strategy are used to control this occurrence. This is commonly achieved by using a more stringent threshold to reject null hypotheses. The Bonferroni correction defines an acceptable global significance level, denoted by alpha asterisk, and each test is individually compared with a value of alpha equals alpha asterisk per meter. This ensures that the family-wise error rate in all M tests is less than or equal to alpha asterisk. When M is large, the Bonferroni correction may be overly conservative. An alternative to the Bonferroni correction is to control the false discovery rate (FDR). The FDR controls the expected proportion of the rejected null hypotheses, the so-called discoveries, that are false incorrect rejections. This procedure ensures that, for independent tests, the false discovery rate is at most Q**. Thus, the FDR is less conservative than the Bonferroni correction and have more power, at the cost of more false positives. Topic: Mis-specification and robustness checks. The main hypothesis being tested, e.g., no association between treatments and outcomes, is often accompanied by other technical assumptions, e.g., about the form of the probability distribution of the outcomes, that are also part of the null hypothesis. When the technical assumptions are violated in practice, then the null may be frequently rejected even if the main hypothesis is true. Such rejections are said to be due to model mis-specification. Verifying whether the outcome of a statistical test does not change when the technical assumptions are slightly altered, so-called robustness checks, is the main way of combating mis-specification. Topic: Model selection criteria. Model criteria selection will select or model that more approximate true model. The Akaika's Information Criterion (AIC) and the Bayesian Information Criterion (BIC) are examples of asymptotically efficient criteria. Topic: Developments and big data. Recent developments have made a large impact on biostatistics. Two important changes have been the ability to collect data on a high-throughput scale, and the ability to perform much more complex analysis using computational techniques. This comes from the development in areas as sequencing technologies, bioinformatics and machine learning in bioinformatics. Topic: use in high-throughput data equals new biomedical technologies like microarrays, next-generation sequencers for genomics, and mass spectrometry for proteomics generate enormous amounts of data, allowing many tests to be performed simultaneously. Careful analysis with biostatistical methods is required to separate the signal from the noise. For example, a microarray could be used to measure many thousands of genes simultaneously, determining which of them have different expression in diseased cells compared to normal cells. However, only a fraction of genes will be differentially expressed. Multicollinearity often occurs in high-throughput biostatistical settings. Due to high intercorrelation between the predictors, such as gene expression levels, the information of one predictor might be contained in another one. It could be that only 5% of the predictors are responsible for 90% of the variability of the response. In such a case, one could apply the biostatistical technique of dimension reduction, for example via principal component analysis. Classical statistical techniques like linear or logistic regression and linear discriminant analysis do not work well for high-dimensional data, i.e. when the number of observations n is smaller than the number of features or predictors p, n greater than p. In cases of high dimensionality, one should always consider an independent validation test set and the corresponding residual sum of squares (RSS) and R2 of the validation test set, not those of the training set. Often, it is useful to pool information from multiple predictors together. For example, gene set enrichment analysis (GSEA) considers the perturbation of whole functionally related gene sets rather than of single genes. These gene sets might be known biochemical pathways or otherwise functionally related genes. The advantage of this approach is that it is more robust. It is more likely that a single gene is found to be falsely perturbed than it is that a whole pathway is falsely perturbed. Furthermore, one can integrate the accumulated knowledge about biochemical pathways like the JAK-STAT signaling pathway using this approach. Topic: Bioinformatics advances in databases, data mining, and biological interpretation. The development of biological databases enables storage and management of biological data with the possibility of ensuring access for users around the world. They are useful for researchers depositing data, retrieve information and files raw or processed originated from other experiments, or indexing scientific articles, as PubMed. Another possibility is search for the desired term, a gene, a protein, a disease, an organism, and so on, and check all results related to this search. There are databases dedicated to SNPs, DBSNP, the knowledge on genes characterization and their pathways (KEG), and the description of gene function, classifying it by cellular component, molecular function and biological process, gene ontology. In addition to databases that contain specific molecular information, there are others that are ample in the sense that they store information about an organism or group of organisms. As an example of a database directed towards just one organism, but that contains lots of data about it, is the Arabidopsis thaliana Genetic and Molecular Database (TARE). Phytosome, in turn, stores the assemblies and annotation files of dozens of plant genomes, also containing visualization and analysis tools. Moreover, there is an interconnection between some databases in the information exchange sharing, and a major initiative was the International Nucleotide Sequence Database Collaboration (INSDC), which relates data from DDBJ, EMBL-EBI, and NCBI. Nowadays, increase in size and complexity of molecular datasets leads to use of powerful statistical methods provided by computer science algorithms, which are developed by machine learning area. Therefore, data mining and machine learning allow detection of patterns in data with a complex structure, as biological ones, by using methods of supervised and unsupervised learning, regression, detection of clusters and association rule mining, among others. To indicate some of them, self-organizing maps and k-means are examples of cluster algorithms, neural networks implementation and support vector machines models are examples of common machine learning algorithms. Collaborative work among molecular biologists, bioinformaticians, statisticians and computer scientists is important to perform an experiment correctly, going from planning, passing through data generation and analysis, and ending with biological interpretation of the results. Topic: Use of computationally intensive methods. On the other hand, the advent of modern computer technology and relatively cheap computing resources have enabled computer-intensive biostatistical methods like bootstrapping and resampling methods. In recent times, random forests have gained popularity as a method for performing statistical classification. Random forest techniques generate a panel of decision trees. Decision trees have the advantage that you can draw them and interpret them even with a basic understanding of mathematics and statistics. Random forests have thus been used for clinical decision support systems. Topic: Applications. Public health, including epidemiology, health services research, nutrition, environmental health and health care policy and management. In these medicine contents, it's important to consider the design and analysis of the clinical trials. As one example, there is the assessment of severity state of a patient with a prognosis of an outcome of a disease. With new technologies and genetics knowledge, biostatistics are now also used for systems medicine, which consists in a more personalized medicine. For this, is made integration of data from different sources, including conventional patient data, clinico-pathological parameters, molecular and genetic data as well as data generated by additional new omics technologies. Quantitative genetics. The study of population genetics and statistical genetics in order to link variation in genotype with a variation in phenotype. In other words, it is desirable to discover the genetic basis of a measurable trait, a quantitative trait, that is under polygenic control. A genome region that is responsible for a continuous trait is called quantitative trait locus (QTL). The study of QTLs become feasible by using molecular markers and measuring traits in populations, but their mapping needs the obtaining of a population from an experimental crossing, like an F2 or recombinant inbred strains, lines RILs. To scan for QTLs regions in a genome, a gene map based on linkage have to be built. Some of the best-known QTL mapping algorithms are interval mapping, composite interval mapping, and multiple interval mapping. However, QTL mapping resolution is impaired by the amount of recombination assayed, a problem for species in which it is difficult to obtain large offspring. Furthermore, allele diversity is restricted to individuals originated from contrasting parents, which limits studies of allele diversity when we have a panel of individuals representing a natural population. For this reason, the genome-wide association study was proposed in order to identify QTLs based on linkage disequilibrium, that is the non-random association between traits and molecular markers. It was leveraged by the development of high-throughput SNP genotyping. In animal and plant breeding, the use of markers in selection aiming for breeding, mainly the molecular ones, collaborated to the development of marker-assisted selection. While QTL mapping is limited to resolution, GWAS does not have enough power when rare variants of small effect that are also influenced by environment. So, the concept of genomic selection (GS) arises in order to use all molecular markers in the selection and allow the prediction of the performance of candidates in this selection. The proposal is to genotype and phenotype a training population, develop a model that can obtain the genomic estimated breeding values (GEBVs) of individuals belonging to a genotyped and but not phenotyped population, called testing population. This kind of study could also include a validation population, thinking in the concept of cross-validation, in which the real phenotype results measured in this population are compared with the phenotype results based on the prediction, what used to check the accuracy of the model. As a summary, some points about the application of quantitative genetics are: this has been used in agriculture to improve crops, plant breeding, and livestock, animal breeding. In biomedical research, this work can assist in finding candidates gene alleles that can cause or influence predisposition to diseases in human genetics. Topic: Expression data. Studies for differential expression of genes from RNA-seq data, as for RT-qPCR and microarrays, demands comparison of conditions. The goal is to identify genes which have a significant change in abundance between different conditions. Then, experiments are designed appropriately, with replicates for each condition, treatment, randomization and blocking, when necessary. In RNA-seq, the quantification of expression uses the information of mapped reads that are summarized in some genetic unit, as exons that are part of a gene sequence. As microarray results can be approximated by a normal distribution, RNA-seq counts data are better explained by other distributions. The first used distribution was the Poisson one, but it underestimates the sample error, leading to false positives. Currently, biological variation is considered by methods that estimate a dispersion parameter of a negative binomial distribution. Generalized linear models are used to perform the tests for statistical significance, and as the number of genes is high, multiple tests correction have to be considered. Some examples of other analysis on genomics data comes from microarray or proteomics experiments, often concerning diseases or disease stages. Topic: Other studies. Ecology, ecological forecasting, biological sequence analysis, systems biology for gene network inference or pathways analysis, population dynamics, especially in regards to fisheries science, phylogenetics and evolution. Topic: Tools. There are a lot of tools that can be used to do statistical analysis in biological data. Most of them are useful in other areas of knowledge, covering a large number of applications. Here are brief descriptions of some of them. SiteDesign, a computer package developed by VSNI that helps the researchers create experimental designs and analyze data coming from a design present in one of 3 classes handled by SiteDesign. These classes are resolvable, non-resolvable, partially replicated and crossover designs. It includes less used designs, the Latinized ones, as T-Latinized design. SAS, a data analysis software widely used, going through universities, services and industry. Developed by a company with the same name, SAS Institute, it uses SAS language for programming. R, an open-source environment and programming language dedicated to statistical computing and graphics. It is an implementation of S language maintained by CRAN. In addition to its functions to read data tables, take descriptive statistics, develop and evaluate models, its repository contains packages developed by researchers around the world. This allows the development of functions written to deal with the statistical analysis of data that comes from specific applications. In the case of bioinformatics, for example, there are packages located in the main repository CRAN and in others, as Bioconductor. It is also possible to use packages under development that are shared in hosting services as GitHub. ASREML, another software developed by VSNI that can be used also in our environment as a package. It is developed to estimate variance components under a general linear mixed model using restricted maximum likelihood (REML). Models with fixed effects and random effects and nested or crossed ones are allowed. Gives the possibility to investigate different variance-covariance matrix structures. Weka, a Java software for machine learning and data mining, including tools and methods for visualization, clustering, regression, association rule, and classification. There are tools for cross-validation, bootstrapping and a module of algorithm comparison. Weka also can be run in other programming languages as Perl or R. Orange, a programming interface for high-level data processing, data mining and data visualization. Include tools for gene expression and genomics. Topic: Scope and training programs. Almost all educational programs in biostatistics are at postgraduate level. They are most often found in schools of public health, affiliated with schools of medicine, forestry, or agriculture, or as a focus of application in departments of statistics. In the United States, where several universities have dedicated biostatistics departments, many other top-tier universities integrate biostatistics faculty into statistics or other departments, such as epidemiology. Thus, departments carrying the name biostatistics may exist under quite different structures. For instance, relatively new biostatistics departments have been founded with a focus on bioinformatics and computational biology, whereas older departments, typically affiliated with schools of public health, will have more traditional lines of research involving epidemiological studies and clinical trials as well as bioinformatics. In larger universities around the world, where both a statistics and a biostatistics department exist, the degree of integration between the two departments may range from the bare minimum to very close collaboration. In general, the difference between a statistics program and a biostatistics program is twofold. Statistics departments will often host theoretical, methodological research which are less common in biostatistics programs, and statistics departments have lines of research that may include biomedical applications but also other areas such as industry, quality control, business and economics, and biological areas other than medicine. Topic: Specialized journals. Biostatistics, International Journal of Biostatistics, Journal of Epidemiology and Biostatistics, Epidemiology, Biostatistics and Public Health, Biometrics, Biometrica, Biometrical Journal, Communications in Biometry and Crop Science, Statistical Applications in Genetics and Molecular Biology, Statistical Methods in Medical Research, Pharmaceutical Statistics, Statistics in Medicine, Annals of Biometrics and Biostatistics. Current research in biostatistics. American Journal of Biostatistics. Topic. See also: Bioinformatics, Epidemiological method, Epidemiology, Group size measures, Health indicator, List of biostatistics journals, Mathematical and theoretical biology.